Hi guys, this is 101 Class Survival Guide, and in this video I'll be discussing about the old methods for nucleic acid sequencing. In the lecture 1, there will only be old methods, so let's get into the video. The contents are simple: what is sequencing and why we do it, and then there are three methods: Sanger sequencing, automated Sanger sequencing, and pyrosequencing. We'll be discussing all these three in detail. Now let's discuss about sequencing and its requirement. The importance of DNA is that it is the code of life and it consists of all the information required to transcribe and translate important biological information. When we analyze the DNA, we get to know more about the organism from which it was extracted, and sequencing plays a vital role in that it provides us with the primary structure of the DNA, which can be further used in experimental analysis. Benefits and uses of nucleic acid sequencing are in the field of molecular biology for studying the genome itself, in which we study how and why proteins are made, what type of proteins are made, identifying new genes, association of genes with diseases and phenotypes, and potential drug target detections. Evolutionary biology study in which we study how different organisms are related and how they evolved. In metagenomics, we analyze the DNA from different abiotic environmental components like water bodies, dirt, air, and even swab samples of organisms for forensic analysis. Genetic testing to detect genes for medical purposes. Sanger sequencing was the first ever sequencing method developed to analyze nucleic acids. Sanger sequencing, which is also known as the chain termination method, was developed by Frederick Sanger and his colleagues in 1977. Sanger sequencing components include single-stranded DNA template, which is achieved by elevated temperature in which the double-stranded DNA dissociates into single-stranded DNA templates, DNA primers, and the clean-out fragment, which is the DNA polymerase I isolated from E. coli, but it only exhibits the 5' to 3' polymerase activity and 3' to 5' exonuclease or proofreading activity, but it lacks the 5' to 3' exonuclease activity. So that is why it is known as the clean-off fragment and not just DNA polymerase I. Normal dNTPs, which are ATGC, and modified ddNTPs, which are modified so that the chain the chain elongation terminates as soon as a ddNTP is attached. Alright, so here we have 4 test tubes in which we have added the same DNA sample or genomic DNA sample, and we have added all the components including the clean-off fragment and the simple dNTPs. However, the difference is that in each test tube we add a different type of ddNTP. In the first one we add ATP, in the second one we add CTP or DDCTP, in the third one we add GTP, and in the fourth one we add TTP. The difference is that the difference between the normal dNTPs and ddNTPs is that the ddNTP lacks a 3' hydroxyl group. The 3' hydroxyl group absent stops the elongation of the chain, and whenever a ddNTP is attached, there is no more elongation and the chain terminates there. However, if a normal dNTP attaches, then the chain termination does not occur and the chain elongates fully. For example, in the first test tube we got two different lengths. Since we only had ADPs attached, so wherever there will be an A inside the chain and a simple dNTP attaches, for example DATP, then the chain can elongate because the dNTPs consist of a hydroxyl group that helps in elongation. But whenever the ATP attaches, it stops the elongation. So we get two different lengths from test tube 1. From 2 we get 4 different lengths. From 3 we get 3 different, and in 4 we get 2 different lengths. So now that we know the basic concept of the Sanger sequencing, let us discuss the steps one by one. The DNA to be sequenced is incubated with the clean-off fragment of DNA polymerase I, a suitable primer, and the 4-deoxynucleoside triphosphates. Either at least one dNTP or the primer is 32P labeled. In addition, a small amount of the 2,3-dideoxynucleoside triphosphates or ddNTPs of one of the bases is added to each one of the reaction mixture. With the addition of the enzyme DNA polymerase, which is the clean-off fragment in this case, the primer is extended until a ddNTP is encountered. But if a ddNTP is not encountered, the chain elongates fully. The chain will end with the incorporation of the ddNTP. With the proper TNTP-ddNTP ratio, the chain will terminate throughout the length of the template. All the terminated chains will end in the ddNTP added to that reaction. After we obtain the 4 different reaction mixtures in which 4 different ddNTPs were added, we run the polyacrylamide gel electrophoresis and the results are somewhat like this. The polyacrylamide gel electrophoresis is not similar to agarose gel electrophoresis. This will be discussed in another video. And the fragments are detected using radioactive markers and autoradiography. However, we can also manually read the gel. For example, in this gel, for example, if these are 4 different mixtures from 4 different vials or samples, then the shortest sample will travel the farthest. For example, in the ddATP containing vial, this sample was the shortest and it traveled the farthest. And the shortest sample was GGC, this one. And it traveled the farthest among all the other samples. So what the benefit is that we get a linear sequence and all the sequences are terminated at a ddNTP. For example, in the first sample the end is ddCTP and in its second there is also a ddCTP in its end. After that comes the end ddATP. So we know that from bottom up there is CCA. This is how we read from bottom up the agarose gel results of Sanger sequencing. This image makes us more clear on how we manually read the sequencing results of the Sanger sequencing method. In the first sample, that is the lowest sample, is usually consisting of only one nucleic acid, such as the farthest here is the T. We know that we also need to note down in which order we have added the samples of our vials. For example, if these are ATGC containing vials, then we must know that this was from the first vial, this was from the second, third and fourth, so that we know which ddNTP was added to it, since all the ddNTPs are attached at the end of each sequence formed. And above it is A. So we know that after T comes A. So the sequence is TA. Then comes G here, G. TAG. Then we have C. TAGC. TAGCT. And similarly we go on until we have all the sequence, which is TAGC, TGA, TA, CGAG. Alright, that's it for this video. Thank you for watching. Please subscribe for more and comment below for any queries or any future requests. The automated Sanger sequencing and pyro sequencing methods will be described in the upcoming videos.